Okay, everybody, time to start the meeting. Come in, get your seat. Yeah, you can do that.
Oh, yeah.
All right. I'm Richard Romano, the Prime Minister, so I get to introduce these meetings. This is the business portion before we have our speaker talking. And so we do the club stuff, which is primarily checking in with Kitty, who does the long distance forays, and Dana, who does the local forays, and any other minister who might have stuff happening, including Les over there. He's on the fair committee for the fungus fair this year. Anyway, so couple things. First off, we're going to have our election next month, and it's only people that attend here that are going to vote. We don't do off-site voting or anything like that. It's always been that way, okay? And I've heard from Dano that probably this is going to be the last Zoom or, you know, hookup for the meetings. They're all going to be in person from now on. And are we still recording them and sending them onto YouTube?
Yeah, I believe the plan will be to get a good quality recording of the speakers, if it's somebody that people want to see, and have those recordings available on YouTube at a later date. We haven't been getting a lot of participation from the Zoom, and I know the Zoom people are a little frustrated with our audio quality and things. So that's why we're doing it.
Okay. So we're going to have our election meeting next month. We have several people running for office. We have Mark Gillespie and Delaney, who are kind of interested in the prime ministership. And then we have Deb is looking for a replacement scribe if somebody is interested in doing that. Zach Potter is going to take over for Kitty in propaganda, along with Anna, you know, to share the stuff there. And I think everybody else may be coming back. I don't know what I'm going to do, maybe a minister at large or something like that, but I'm kind of done, you know, come next month with my 2 years. So I'll be sending out a reminder on the Google groups that anybody who wants to nominate somebody, nominate themselves. We're always happy to take people from the floor. There's a lot of good, fun things to do as a minister. And we hope that we'll get more participation by people who really love to do this. In particular, also, we are looking for lots and lots of volunteers. We're going to put on a new, redo the fungus fair this coming January, second full weekend in January, and we're looking for a lot of volunteers. And yeah, well, Les is going to take your name. Anyway, so and we need good people that can manage certain aspects of the fair and just take it on and manage their volunteers and that sort of thing. It'd really be helpful because the fair is quite a multitask sort of thing. Lots and lots of people participate in putting it together and taking it down. It's pretty much a 5-day affair. We start on a Wednesday, we tear down by Sunday evening right after the fair ends at 5 o'clock. And it's always great fun, lots of excitement. It's going to be a little bit smaller this year because we won't have the after dinner or after hours dinner. But we still will have vendors. We'll still have the kids' room. We'll still have the main room for mushroom displays. May not, and Bob's not going to be bringing us delicious cooking, but we will have Penny Ice Creamery and a few other people showing up like they usually do. So it'll be fun. Let's see. Kitty, you want to talk about long distance forage? Oh, you have a question? You mean in the Oh, the reason is we don't have anybody to run it. Yes. Who's? If you take it on, but you were bowing out. Okay. Okay. Fine.
Cheers back on.
Last I heard
That's right.
fine. I don't care. I'm more than happy to be flexible. Anyway, so we'll be after hours dinner. That's fine. That's great. See, the problem with the fair has always been we have to have really dedicated people to take on sections of the fair because not everybody can, do the whole thing. We lost quite a few members this last year and a half, and with that, we need, people that are willing.
I know very responsible people that are willing to step in and help by doing that.
He'll start you on that. Great. Okay, super dudes. That's what we need is people that are willing to step up and commit to doing that and organizing it and that sort of thing. Yes.
So as long distance foray minister, hi, I'm Kitty. You can hear my voice. Oh, Dan, stop, take me away. As far as the forays for the rest of the year, there is a private foray going up to Alaska this summer, but it's all full. There's a wait list. As far as morels and spring kings, it's going to be on the fly. It's not going to be a formal foray. We're going to, when the snow melts, the logs are off the road, the snow is off the road, and we have access to an area, we will post the date, the location, and people just show up. So that's what our summer foray is going to look like.
That's our good old days. That's what we used to do.
Because we don't know when that snow is going to melt.
And where will this be posted?
It will be posted on Google Groups, Facebook, and our webpage.
All right.
So, pie, but I'm not even sure we're going to do it at pie. It all depends on what's open, what's available.
So the dates for last one.
Yes.
It's done.
It's done.
It's done. You missed it. You were in Spain. It's gone.
I missed what you were talking about when you started.
No, that's for the
Morels.
Morels and spring kings. Okay.
It may not be at Pipeline. It will be wherever it is.
Heads up for the following forays for Albion. Albion this year, we had it in December last year, and it was a little late. So what I'm doing is I'm guessing that I'm moving it up earlier. So it's going to be in November. So it's going to be a little earlier, back to what we used to have. So I'm kind of betting on that. And then the January one is still about the same time frame. So it'll be after the fair.
Isn't there didn't David Aurora usually do something on the weekend by Thanksgiving?
Oh, yeah.
Oh, okay. All right. Anyway, so long distance forays. On another note, the Suds and Shrooms this year will be at Peter Moo's house in Romano. And that'll happen on the 20th of May. And Peter will give directions and all that sort of good stuff as to how to get to his place. He's right off the 101.
Not only is he on 101, he's sitting on top of a hill, and the vistas are absolutely spectacular.
Right. So we couldn't find a venue in Scotts Valley. Siltanen doesn't work. McDorsa was locked up. So we had to improvise, and Peter graciously offered his place, which has lots of vistas. He's got barbecues. He's got plenty of places for people to, wander around and enjoy the scenery. So yeah, that's kind of cool. That's our last major event. Suds and Shrooms. We're going to have the Zymer geeks there, and people bring appetizers, food, stuff like that. Potluck. Yeah, exactly. So, and now, yeah, we'll get to him. I'm just making sure that we cover all the bases here. Then now we have Dano, who's our local 4-H leader, and he will bring us up to what's going on here.
Oh, actually, nothing's going on. I haven't done it in local 4-H this spring because it's either been too much rain or then we hit that sort of dead spot in the middle, and then I was tied up with other events. But there are mushrooms out. There aren't really enough mushrooms to lead a formal 4-H because, you know, I'll walk 15 miles and find one patch of chanterelles, which is good for me, but not really that good for a group. But there are patches of chanterelles still out on both sides of the hill. The Amanita villosa have been having a pretty decent year. I've been finding them sporadically. So just keep getting out into the woods, and hopefully next fall there'll be more going on.
Anyway, so I think unless there's, Margaret, did you have anything you wanted to add? No? Okay. And so now we have Les. He's the last of the greats. I've done it already. But you can do it much better.
Okay. Let me see what we got. All right, half of you are new. How many new members this year? Any? Okay. How many have been to a London Nelson fair?
How many have been?
Yeah. It's been a few years. How many don't remember? Anyway, we're not going to deal with volunteers tonight. We use about 125 slots for volunteers on fair. But it's a little early to start begging for people. Tonight's a much more exciting opportunity. I'm not going to do this in another year or two. Medically, I can't continue. But the Natural History Museum in Santa Cruz has also stepped back, and they're going to fill some of the management slots, kids' room and publicity and volunteers. We're going back to them. Unfortunately, they're not affiliated with the city anymore, so we don't get free rent. So
we're still doing it at London Nelson.
Yeah.
And just paying it forward.
Yeah. So now's a good time to step up and create new stuff. We're looking at online sales for ticketing instead of the lines out the door. So that's a new thing we're trying. We have new blood coming in. Like I said, the volunteers from the Natural History Museum are going to provide a lot of staffing, but they're still always staff kind of positions. This is the staff list. It's double-sided. It's not a full-time job. You have really tough decisions to make, like how big should the graphic be so it fits all the different size shirts. That's the kind of tough stuff you have to figure out. So I'll talk with you after tonight. But if people want to join in and help run the fair, especially, I'll spend time with anybody who wants to take over one or more people actually organizing the fair. I'll work with whoever they might be, because there's some technology transfer. It's mostly a bunch of files and emails that you have to remember to send out and you get things going again. It's not that crazy. And also the volunteer. Well, the volunteer thing is just begging a lot.
It's got a program that they put there.
Oh yeah, I've got. It's a iVolunteer site. I just copy from last year to this year, and all the slots are filled out. It's really not that hard once you got it going. I'll tell you how hard it was. I got the job about 7 years ago. Sunday night when we closed up the fair, I was stepping off the stage with Margie, who was moving to Oregon. And we're going down the steps and she says, hey, we should get together to talk about the turnover. And then she left and I went, the turnover? And then I found out that I had the job. Okay. Didn't get a lot of documentation, by the way. So, but there's a good record now to help move things along, and it's a lot of make sure you get the trash cans ordered, and make sure you buy the sand, and buy some gaffer's tape and things like that. It's not too crazy.
Get the tables from the.
Well, we have table counts. We have table counts. We have layouts. We have, we've done it enough. 40-something times that we have some idea what to do. Sure. So anybody who wants to step up and help with that, consider picking it up. That would be nice. And I will definitely work with anybody who does that. Sorry. Okay, well. Well, the real work for the fair setup itself is about 3 months, 3 to 4 months. So, but again, it's not a full-time job because I had a full-time job when this happened, and and I can promise you it won't be that way.
But you were a manager, right?
A manager? No, I wasn't a manager. I tried that and got out of that as soon as they asked me to do management stuff. So, I think that wraps it up. And we'll see you January 12, 13, 14.
Yeah. We're doing that. Okay. So what? Yeah, Margaret's the person to see if you want to be a member, if you aren't already a member, or you want to renew. Let's see, Peter, you're going to introduce our speaker, please.
And Peter's
I know you're busy flipping those mycelium cakes, but
if you haven't seen them already, supposed to be chicken breast for everybody to sample later on. So my name is Peter. I'm the program minister for the Fungus Federation. It's my job to invite speakers every month. And I'm trying to, my goal really is to kind of mix it up a little bit. We have the traditional scholars, the PhDs coming from universities and talking to us about what's, in the cutting edge of research. And then we have, mycologists that are in the field, really leading the foray, seeing people out. And then tonight we have Nick, and he Stanford graduate, grew up in Georgia, and he's actually working in industry. His company, Meaty, manufactures these mycelium-based fermentation meat. What do you call it? Just alternative meat. Yeah, so it smells really good. Huh? Yeah, so the spread. Yeah. And so I found out about this last night. He stayed with me for a couple days. I found out about this that they carried in my scraps. So he went over to Palisade this morning, cleaned out the store, and brought them all here tonight. So we're gonna have a tasting through that. So without further ado, I want to introduce Nick.
Thank you so much for the introduction. Thanks to everyone for inviting me here today and coming to listen. I have a lot of great stuff to talk about. And as Peter was saying, we're going to have the meaty in the back. And so just as like a quick disclaimer, I definitely encourage you to try it. It is vegetarian, vegan, animal-free, and cruelty-free. If you want to check out the labels in case you have any allergies, there's some boxes over there. Feel free to read over it just to make sure that there's nothing that you might be allergic to, but it's a really minimal ingredient list. So I think most people should be good to go to try it. And so let me just get set up with my slides here. All right. Okay, so hopefully everyone online can see things here. If you need to, feel free to pop a message in the chat or just kind of come off mute and try and jump in, but hopefully everyone should be able to see this. Try and keep a quick timer on, it's about 7:30. Okay, great. So welcome to my presentation. I'm going to talk to you about fungal biomaterials, which are a very new and exciting field in industry right now. And so I wanted to prepare this talk for the audience of the Fungus Federation. I imagine that we have people with all different types of expertise in mycology, in mushroom hunting, in mushroom cultivation. And so I wanted to try and thread the needle between making a talk that was good for a lay audience, but also for an expert audience, because I imagine a lot of you are experts. So with that, I'll kind of just jump right in. If you all have questions after a slide, after a section, feel free to just raise your hand and I'll do my best to answer it as we go. So let's see. Okay, so first I just wanted to do a quick self-introduction just about my background, what types of things do I work on. And so I've been working in industry for about 4 or 5 years now. I did work at Stanford with some soil fungi, EM and AM fungi that are kind of plant symbionts. However, I recently worked at MycoWorks. You may have heard of them. They're making mushroom leather out of the Ganoderma and reishi fungi. I worked there for several years. I saw the company grow from 10 people to over 250 and really just saw this rapid growth and saw the product evolve. And most recently, I'm now working at Meaty, but I'm an avid mushroom hunter. I've been able to travel in Central America and work with some indigenous populations and do ecology surveys and things like that. And overall, I'm just really passionate about microbiology. So with that, we can kind of get in. I'll start in just talking about these different fungal biomaterials that we see. And so, you know, once again, apologies if any of this is review, but as I said, just trying to give a really broad scope kind of picture here. So as you can see on this chart, there's a lot of interesting photos and there's a lot of interesting products out there. Mycelium is so versatile and people are doing all kinds of crazy and creative things with it, including making styrofoam and a packaging replacement, making building blocks for buildings, as you can see in this huge tower here. That was an artistic display, but it's still like a functional, you know, structure. People are looking into fire retardant sheets, insulation for homes, acoustic paneling for homes, leather and textiles. And you can see this handbag here made out of a mushroom leather. It almost looks indistinguishable from an animal hide. And that's the type of thing that I was working on in my past job. And additionally, we'll talk more about the mycoprotein, the food products. People are creating not only a whole food, but also these food additives that can go in, maybe a bitter blocker, maybe a protein additive that you can mix into another product. And so the properties of mycelium are what allow people to make these incredible materials. And I'm sure I don't need to belabor the point to you all, but the fact that mycelium is so malleable, it will grow into whatever mold that you provide for it. And it can be grown in both a solid and a liquid substrate. So you can obviously do wood chips, sawdust, straw, corn cobs. These are all solid substrates, but you can also take, for example, like beer wort as an example, or any type of liquid broth, and that can be grown. You can throw fungi in that broth as well. So the fact that you can utilize a different substrate, a different container, and a different species, these are all things that we can turn the knobs and wheels to generate a material to match a function or a desired purpose. So, you know, some of these properties I was just listing, all these abiotic factors, temperature, humidity, the rate of air exchange, the culture medium, as I was saying, solid versus liquid, what nutrients do you have available for the organism? And then additionally, the species you're looking at and anything you're doing to that. You can perturb the culture, you can transfer it to another vessel, you can give it more nutrients. All these things can be done to enhance the growth, to get it to grow differently. And these are the types of things that people are exploring through their companies and through their projects. And so here I just have a quick little kind of workflow chart. This is a very basic look, but this is a kind of laying out how do some of these different materials get made in a common fashion. So on the left side, we have in the green, the abbreviation here is, I believe, wild basidiocarp foraging. So you can essentially go to the woods, harvest these mushrooms. And then the classic example here is like the Amadou hat or any product or an artist comp that you turn into a portrait or a painting or something like that. These are just directly pulling from nature. But then you of course have propagation, and you can go to, for example, in the bottom in the red, SSF, solid state fermentation. You can grow mushrooms, you can create spawn, you can grow trays, and they're listing here that you can create a biofoam or a fabric. So once again, the mycelium textiles, mycelium foams are kind of coming into prominence right now. But then additionally, something I'll talk about throughout the evening is a liquid state fermentation. So you kind of see a shake flask, you see this little bioreactor, and that is a novel method that really is not so common, but has some of the most potential because you can get a really high bioefficiency and a high yield through the liquid cultivation. And they have some more examples here, nanopaper structures, you can make paper out of the fungal pulp. So there's such a wide array of these products. And I just want to talk about a few of them to kind of get the conversation going. So I'll talk a little bit more in depth about the properties of mycelium that can lend themselves and how people are leveraging the organism to get these cool results. And so, you know, this might be a review for a lot of you, but when we're growing on a solid substrate, some of the main morphologies you might see are going to be a tomentose hyphae, a rhizomorphic hyphae, or an aerial hyphae. And these are very much, the organism is responding to its environment and it's able to grow in these different ways. So I kind of included these photos to kind of show the difference. We have two aerial plates, and we have a tomentose mycelium on top, and we have a rhizomorphic on the bottom. And you can see the distinct properties, right? Just from looking at it, you have a very wispy and thin colonization on the top. You have this very thick corded hyphae that just kind of appears stronger on the bottom and appears like a very distinct morphology. And so these kind of have different roles for the organism. The tomentose hyphae might be exploring a new ecosystem. They might be exploring a new food source, and they're just there to colonize and establish the whole. Whereas the rhizomorphic hyphae might be more of an established, they might be a nutrient transfer route between two distal parts of the colony. And they can kind of, guide and transfer water or nutrients and things like that. So these are some distinct morphologies. And then the aerial hyphae is one of the most unique forms of the hyphae, and it's also primarily put into certain products. For example, mycelium bacon can be used, made from the aerial hyphae. So it's something that people are leveraging, and these aerial hyphae are going to be growing into the void space. And so whenever, if you have a mushroom, for example, and something that people call fuzzy feet, you have these hyphae coming out of the base of your mushroom, those are going to be aerial hyphae. They're trying to get oxygen for the culture, for the organism. And so these aerial hyphae tend to grow directly into the void off of the substrate. And so this is kind of a diagram of, this isn't even technically a true fungi, but this is like a mycelial bacteria, but I really just want to show the concept of the aerial hyphae. So you essentially have agar in this bottom lens, you have the void space of, say, a petri dish, and all these hyphae that are growing just straight into the void, these are the aerial hyphae, and through these little kind of micrograph photos, you can see the different morphology. It's operating differently, and the same organism has these fully different modes of growth. So people leverage all these types of growth in solid state. And then there's even more types of growth in liquid. So I'll kind of go on to talk about a couple of those. So I think the unique properties of mycelium in liquid is that they're going to respond to the fusion factors that you give them. So once again, you can control the oxygen level in the system, you can control the rate at which you're mixing the liquid, and you can also control the nutrients that are going in for the fungi. When you modify these different growth factors, you can get two main morphologies, I would say, and there's very much a spectrum between them, but you could have a pelleted morphology or a dispersed morphology. And this is something that we can see at a macro scale. If you look at the culture, you can either see that it's pelleted, or you can see that it's fully dispersed and these hyphae are just extending throughout the liquid in a very kind of even manner. So the pelletization is kind of a response to stress. You can imagine if someone dunks you into water, spins you around really fast, you're probably not having a great time. And that is how the fungi feel, and they want to protect themselves. They want to protect their own life and grow in like kind of a safe manner. So they literally form a pellet, and they just kind of extend their hyphae out, but they're very thick. They're very kind of conglomerated in that pelleted form. And then the dispersed mycelium, sometimes called flocked mycelium, it's just dispersed into the liquid. So if you have a slower rate of mixing, if you have a more gentle culture, maybe agitating, maybe a shaker table or something like that, you'll get the hyphae growing evenly throughout it. And these two types of growth have a different nutritional profile. So if you're looking at a medicinal product, if you're looking at a food product, you can control what your organism produces for you, whether you want a lot of cordycepin from cordyceps, or whether you might want some ganaderic acid from your Ganoderma mushroom. You can control the growth and augment the media to match the product, and also augment the growth factors and kind of environmental conditions. So this is kind of just a figure to show visually what I was kind of describing. So we have dispersed mycelium on top, pellets on the bottom, but they can both be reached from the same inoculum. So say that you have a chunk of agar, you throw that into a shake flask, you mix it really fast, you might get a bunch of pellets in there. Take that exact same agar, put it into a shake flask going half this fast, you'll get a much gentler mixing, you'll get this dispersed mycelium. And this is something that people kind of go back and forth on, and there's entire papers looking at one species. How do we make it pelleted? How do we make it dispersed? What controls for that, both on the environmental level, as well as on the genetic and DNA level. And these are some of the investigations people are working on. And then it kind of goes even a little bit deeper. So not that I want to go into too much detail, but essentially, even within the pelletization, you can have these pellets that agglomerate together and kind of link to one another, or you can have these non-agglomerating, non-linked up pellets. They're just distinct little orbs that kind of float around. So I just kind of wanted to highlight all the different types of morphology we see, some examples in solid, some examples in liquid. So maybe I'll just pause here and see, are there any questions so far? Anything that I could maybe go into more depth on that you're curious about? Oh, yeah. Yeah, so I was just reading a little bit about this. This is something that I pulled from a publication that I linked here. And so I'm not super familiar with this class of organism. It's like an actinomycete. And my understanding is that it's a mycelial bacteria. So genetically it's a bacteria, but it grows in a form that's very similar to fungi. So it has hyphae, it forms a mycelial network, and it's one of these organisms that's kind of not easy to bin. Like it's not a full-on bacteria, it's not a full-on fungi. Something like that. I mean, I think the taxonomy is pretty clear for it, but as far as like the physical morphology that we see, it's very similar to fungi, and you kind of get into, is it a true fungi versus not a true fungi and things like that.
Okay. Yeah. Speak up in the back so they can hear you on Zoom.
Yeah, it's super unique, and I would love to study this and learn a little bit more. But just to kind of review the morphology here, you have this mycelium that's growing deep down into the agar, and that's obviously trying to get at the nutrients of the substrate. It wants to consume directly all those carbohydrates, all those nutrients in the agar. And then the aerial hyphae might be trying to just simply oxygenate, but they can also form reproductive structures as well. So a lot of these ascomycete fungi, for example, they might form conidia as their reproductive structure. These aerial hyphae will grow up into the air, try and get more oxygen for the culture. As the culture reaches maturity, they'll develop into these unique sexual structures called conidiophores, and they'll eventually be able to kind of just shoot their genetic material in the form of these conidia out into the wind and kind of propagate. So it's a super unique morphology, and it's something that people try to dial in and control, because if you can control the morphology, you control your end product better. And thinking about industry, thinking about companies that want to create a scalable product, you want the consistency, but the biology is what's getting in the way. And so it's all about kind of trying to tame this very unwieldy organism, unwieldy system, and get it to do exactly what you want. Yeah.
So are there preferential forms of the mycelium that are, enhanced as far as nutritive value, as opposed to mycelium that are structurally made to produce a material that is not consumed as well, like, as you say, leather, or structural building materials, or styrofoam, things like that, which is non-nutritive in its basic form. I just wondered, is there a preferential way of producing a mycelium that enhances its nutritive aspects?
The answer is definitely yes. And I think it would something you might want to dive into, maybe case by case, or once again, kind of species by species, They all have their distinct nutritional requirements. And for example, you might need a certain concentration of copper for a given organism. Then you take another species and it wants a different amount and it has a different profile. And so I think it really does depend on the organism in particular, which isn't necessarily a super satisfying answer. But additionally, what I can say is, having worked in the industry for a long time, when we're making these products out of mycelium, it's kind of you're racing against the clock because you don't want it to reproduce for the most part. And so in the example of growing the Ganoderma mushroom at MycoWorks, we never wanted to allow it to reproduce because it would ruin the product, and we just wanted the mycelium. And so as soon as it starts to make a fruiting body, it's discolored, it has a different texture, and it's just kind of an anomaly that we can't factor for. We can't consistently get it to fruit every time. So instead, we're going to consistently get it to remain in the vegetative stage. And so I think a lot of companies just try and target that vegetative growth, take it as long as they can without it going to reproduction, or do everything to mitigate any triggers to reproduce, and then harvest it and on.
Economics are actively leveraged in growing in compost.
Oh, no way.
Yeah,
so it's like a pre-treatment step before inoculating, or
up in there they grow, they have their time. But it's fun.
That's super interesting. All right, so a quick little just visual interlude here. I just wanted to show some photos that I've taken doing some fluorescent microscopy, and this is work that I was doing at my current company, kind of in the last year or so. But just kind of showing that this is mycelium that was grown in liquid, and what you can see is that it's forming a pretty normal type of structure. For me, this kind of looks analogous if I took a sample of something that had been on a plate, for example, or if I took a sample from something that had been in the soil. It might look very similarly. Obviously, you have to go through the process of plating it and getting it into liquid to visualize it under the scope. But I just think it's so interesting, and something that we're kind of diving into in the industry and at the company I work is that we want to get really into the cell level and understand what's happening with the growth, because there's cell wall components that contribute to the texture of our final product and food products. There are concentrations of nutrients that we want to understand. Is it limiting? Is there a part of the fungal cell that maybe is underdeveloped or something like that? And so by utilizing these fluorescent dyes, you can not only get these really amazing kind of artistic quality photos, but we can also learn about the actual cell structure and kind of the intricacies of the cell interactions. So these are just some fungal hyphae once again. It's interesting to see we're often using a stain that's targeting a particular organelle or a particular part of the cell. So there's stains that will stain the chitin. There's stains that will just go and blanket stain any DNA that it can get its hands on. And there's stains that are even more specific. So we can kind of leverage these and even two stains in combination to get a desired visualization of the hyphae. And here's just one more photo, seeing these interesting stains. We are really interested in the morphology as well, because it can really indicate stress as well. And so we don't often think about if the mushroom is stressed or not, or in the case of, say, mushroom farming, you obviously want your mushrooms to fruit, but are you really checking in and saying, is that mushroom the happiest it could be? That might be not even worthwhile to think about. But if for something as delicate as the work that we do at Meaty, we actually need to ask those questions and say, how happy is it? How aggravated is it? Because it can produce an off flavor that you can taste in the food. And so it's a matter of keeping them happy, keeping them in a delicate state, really babying it in a way, but that's going to give us our best product.
So this would be considered pelleted, right?
Not exactly. So what we're seeing is like the hyphal branch just kind of extending here, and each circle is just a cell. And so there are some areas where the hyphae is kind of really bunched up together, but maybe jumping back to an earlier photo, some of it might be an artifact of how it was placed on the slide. But this, for example, I would call this like a pretty dispersed growth overall. You can see a little bit, once again, it's kind of a collaboration, but in the grand scheme, like it could be much worse. If it was truly pelleted, this entire area might be okay. You might not even be able to see through because there's just like tens upon tens of layers of hyphae, all just interspersed. So this is more on the dispersed side. Yeah. Checking the chat here. Oh, no questions so far. Cool. All right, so I'll kind of switch gears here, or taking a kind of tonal shift, and talk a little bit more about industrial fermentation. What's the state of the industry? What really is going on at a large scale with regards to fermentation? So at a really broad level, industrial fermentation is just anything that's a fermentation at scale to produce a product. So this really encompasses a wide range of industries and a range of products that we really see every day. So just some basic examples to throw out. We have food and drink. You have kombucha that's fermented, cheese, bread, beer, and wine. The list goes on. But we also have medicines and specific metabolites. So things like penicillin, vaccines, antibodies, discrete components of a pharmaceutical final product are all produced with fermentation. And then additionally, metabolites, things like vitamins, acids, ethanol, clostatin, all these compounds can be produced through fermentation. And it's by that precision of knowing the organism you need to grow, knowing your target molecule, target end goal, and just gearing the fermentation to get the best amount of that final product and the best quality of that product as well. So it's obviously so common to see something like bread or beer, but it's not as common to think about, oh, right, all these vitamins that I'm taking, oh, all these vaccines that are being mass produced and sent out to the world, they can often be the product of some kind of microbial fermentation. And drug discovery and novel compound discovery is also a really exciting part of the industry as well. So my kind of financial stat here is that microbially produced biopharmaceuticals had a revenue of over 100 billion in 2017 alone. This is an absolutely massive industry, and it's something that is not really well known in the mainstream, where you don't really hear about all these different fermentation companies that are working. There's not terribly many of them, but it's obviously a huge monetary value. And the novel compound discovery I was mentioning is a huge part. So how do we get from our organisms in the forest to culturing it in a lab to knowing what to extract from that to make a medicine or to make a cancer treatment or to make a medicinal product that's going to elevate your quality of life and your quality of health? It's a matter of going to the DNA level. It's a matter of using predictive modeling. Some companies are using AI to generate and to screen the DNA libraries that we have to generate these new compounds. But all of this stuff, the eventual end goal is like, we want to ferment that microbe, extract the potent compound, move on with our day and make our product. So it's just this interesting connection where you have your macro scale nature, then you have micro scale DNA level kind of work to do to really understand some of these compounds. So that's just a little bit of an overview, and I kind of want to dive more into some of these differences. So I kind of mentioned that these biotech companies are using a solid substrate, some companies are using the liquid substrate. What really are the differences between those two processes, and why can't I make the same product using either? And the answer is like kind of more nuanced than you might think. So once again, just kind of a lot of text on the screen. I'll try and kind of just make it really brief, but for a solid state fermentation, kind of mention some examples. If you're growing some oyster mushrooms on grain or sawdust, that's a solid state fermentation because your substrate is solid. You're usually either going to pasteurize or sterilize your substrate. You're going to try and inoculate in a sterile fashion. You're going to get your fermentation to occur, and then you're finally going to harvest that finished product, whether that's a mushroom, whether that's a myceliated grain, or whether that's a biomaterial like a leather or a building block. Those can all be made using solid state. And then liquid state is kind of just a little bit of a different scenario. You have brewing beer or wine as kind of classic examples of this. You're inoculating your kind of sterilized liquid broth with your target yeast, and that's going to give you your alcoholic beverage. And so you always have your nutritive broth. You can generate a liquid culture. You can take that and put it back onto a solid state that some people do to cultivate their mushrooms. And it's not as common, and not as many people are, at least at the casual and kind of hobbyist and home cultivation level. Liquid fermentation is not nearly as common because it is more challenging, but it can be more efficient, and you can create a much more specialized product depending on how you do it. So we'll kind of talk more about that as we go here. Just kind of wanted to lay it out more visually and kind of have like a couple workflows built into here. This is really a little bit directed at mushroom growing, but it can give you an idea of what steps would you go through, not only at a home scale, but also at an industrial scale. A lot of these principles are conserved. A lot of these techniques and methods you're doing are similarly. And so for a solid state workflow, it's kind of straightforward. You're going to have your starting culture in the form of a master stock. You might have an agar plate. You might have a liquid spore culture. You might have a spore print. You're going to want to wake that up from storage. You're going to want to grow it onto agar. You're going to want to take it to a spawning and a nutritive substrate in the form of something like grain spawn, very commonly used. Then you're going to allow your grain to colonize. You're going to go to your kind of fruiting substrate, and whether that's going to be sawdust, straw, other byproducts of agriculture. Always doing these kind of sterile inoculations, sterilize the substrate, do a sterile inoculation in some kind of a clean airflow. And then you finally are going to go to that final step. And that might be, once again, harvesting your mushrooms, harvesting your leather, harvesting your mycelium bacon. With a lot of these biomaterials, there's post-processing after the fact. So not only are you growing this product, but now you have to turn it into a food product, you have to turn it into a leather, maybe you have to dry it, maybe you have to treat it with a special way. But the basic principles are the same. You're going from one solid substrate to the next, iterative transfers, doing some percentage of inoculation that's going to expand the culture, and you're going to get your final product. And I just wanted to contrast that with the liquid state workflow, which once again, some things are similar and some things are very different. So we're once again starting with our master stock, agar plate, spores, or liquid culture. Now, in the case of liquid fermentation, you really want to be careful about your media formula. And that's to say, what are you putting in the broth? What nutrients are you putting in there? Do you want to use something like malt extract that might have a lot of different nutrients in it, but you don't know the exact concentrations of each of them? That would be an example of like a complex medium, because there's so much going on and you don't know exactly. And then there's an example of a defined medium. So do you want to know everything that's in there? Did you add the chemical elements one by one at a particular weight so that you know, I have X grams of copper in here, I have X grams of zinc, I have X grams of magnesium, and that's as much as my culture needs. So there's kind of a lot of debate about that. A lot of companies love the defined media, but the complex often gives a better result because it has more nutrients and you might not be limited. But then you also don't get the consistency. So there's so much push and pull there. But all that being said, back to the workflow, you're going to have your liquid media. You're going to inoculate that in a sterile fashion. You're going to want to agitate that culture, because if you just leave something sitting with no agitation, it's not getting oxygen and it'll eventually run out of oxygen very quickly in that liquid. So you want to stir it around, shake it around. There's entire machinery devoted to that mixing step. You get your mature liquid culture out the other side. And now it gets really interesting because you can do so many things with this. You can take this liquid culture and put it back onto a solid substrate, as I was saying, go onto sawdust, go onto grain, but you can also take that, transfer into a larger body of liquid, and then eventually harvest the mycelium from there. And so my classic example here, it's such a high value product, and there's a lot of studies going on to the medicinal efficacy. We have cordycepin, a very kind of important component of why cordyceps has such a potent medicinal effect on the human body. And that's something that people target when they do liquid fermentation, because you can try and upregulate and just say, I want to grow cordyceps, I'm going to grow it in liquid, and I want to get as much cordycepin as I can, as much bang for my buck, so that I get a good harvest and a good yield. So it's just two different kind of styles of fermentation. They both have pros and cons, and I even have them listed. So there's some kind of basic, comparisons here. So the solid state fermentation, you often have a cheap substrate, as in the form of ag waste, forestry waste. Liquid state fermentation, you're often making that media yourself with these powderized chemicals, crystalline chemicals. So it's just a different method of substrate preparation. For solid state, it can be really low tech. You can do outdoor pasteurization. You can do a still air box for inoculation if you're at small scale. All these things that don't require specialized machinery. However, in liquid state, you can't really get away with pasteurization. It's a broth of liquid with sugar in it. Every single microbe that is alive wants to grow in there. So you need to sterilize it. You need to keep it clean, and it needs to be in some kind of an airtight or at least a cell-tight container. For example, using a micron filter that no cell can pass through, but the oxygen can pass through. So the bioefficiency of solid state is not off the charts. People do get good values, and it's kind of just been the standard for so long. But with the liquid state fermentation, you can dial in your media so that you don't have any extra stuff in there. If you consume all your sugar and all your trace metals, you're left with just kind of like a water at the end, and then you have your product. So you can dial this in, but it's very challenging to know what concentrations to use. And then finally, solid state can be done indoors, outdoors, in between. You can cultivate mushrooms in your garden. You can grow mycelium in your house. The liquid state, it's not as forgiving. You're usually seeing it indoors in a lab or a factory because you need all this special equipment. So it's nowhere near as accessible, but in the industrial space, in the biotech, in the startup space, it's really appealing because it's more scalable than solid. And you can go to these huge reactors. I think Corn held the world record for the largest reactor. Maybe they still do. And it's like 250,000 liters or something absolutely insane. You look at like a grain silo. It's like a bioreactor the size of that, just towering into the sky. And that is just churning out mycelium day after day. And so we'll kind of look at a diagram of that later on and kind of talk more about that, but it's a super unique process and just really amazing what we've been able to innovate and do. So I talked about a lot of kind of techniques. I talked about some workflows, and I want to move on to biomaterials. But before I do, I'll just take another pause, feel the room. Any more questions? Anyone have something they're curious about right now? Yeah.
So a lot of work has been done on psychoactives. What company or anybody applies these kinds of things to when the get something that could help somebody to maybe or whatever?
Yeah, that's a super good point. So right now, I can say that my company is not doing anything in that space at this particular moment. We're kind of focusing on food and just absolutely trying to scale as best we can and grow as rapidly as we can to make our food product. There is a company, Mycelium, who does do more in the realm of the psychedelic research. And I think that they've done collaborations with other companies. I believe that they have even a couple locations around the world. They have like a site in Jamaica. At one point, they had a site in Colorado. So I think there are companies working on that. But I think what I would say to your point as well is just there's really a lot more to learn. And looking at something like psilocybin mushrooms or any other kind of, even like something like a cannabis plant, if you're giving this organism the best nutrition, you're going to get the best product. And the testing for things like psilocybin hasn't really been normalized. We don't really know what's our concentration, what are all these different kind of compounds that are going on in this mushroom that I'm eating and obviously having an effect. But I think as the testing gets better, we can link together the substrate and the nutrition to the final product and the concentration of these active compounds that are so beneficial and therapeutic. So I think that's a bridge that we can build, and that is starting to be built now. Yeah.
Are the waste products from the bioreactor, like apples belch? What do you do?
Right. So for the bioreactor, I can only kind of speak to the systems that I've worked in, but there's really not a huge amount of waste. I think the primary waste is like the spent media itself, and that's kind of going to be the leftover broth that just has a little bit of sugar in there and some trace elements and things like that. And it's by no means toxic, but it's also not something you just want to be dumping into a river or something like that. So finding kind of the proper wastewater treatment can be challenging. A lot of companies try and create a more circular loop where they can either reuse that spent media or turn that into a different fermentation kind of workflow. So the waste is not really crazy. And I think that we have done kind of carbon footprint analysis, comparing something like meaty to something like a large scale cattle farm. And I think it's just like no competition. It's just clearly an advantage for the fermentation. It's all indoors. It's kind of vertically scalable. And so for all these reasons, and the inputs are minimal. It's obviously like water and electricity, but just for what we get out of it, compared to something like cattle farming, it's just not even close.
Yeah, you are going to produce CO2, but not in any larger quantity than what the raw materials would consume when they're grown.
Right.
So you, in your product, which is a food product, are you optimizing for like a protein substitution or a protein substitution with a certain mix of minerals and vitamins and things like that? Or are you just, still experimenting with the mix, as it were, to replicate the nutritive value of what it is that you consume? Because I read the crossword puzzle that the only non-meat source of vitamin D is mushrooms.
Yes. So that's a great point, and I have several answers to give you for that. So first and foremost, I think right now we do, solid nutritional testing of our product. The things that we know and feel confident about is that it is a complete protein. It has a diverse suite of amino acids. I want to say like at least 9 different amino acids. And we also are able to kind of impart vitamin D into the food itself, which I can kind of say more about that. At the moment, we're not optimizing for any one of those things in particular. I think we feel good about our media formulation to where we're giving the organism, for the most part, everything it needs. It's really hard to exhaustively say like, Are we out of zinc right now? Are we out of magnesium? Are we out of copper? Are we out of all these different, like, just kind of trace elements, and even the other things as well, like nitrogen, amino acids for the organism to consume. And so it's hard to do that testing to get that fine-grained analysis, but I think based on our media formulation, we've kind of taken a very old school recipe and kind of updated it for the modern day and done our own tweaking and done our own testing and things like that. So I think we've gotten into a very good state. Is it perfect? No. But are we making a very nutritious and healthy food with it? Definitely yes. And for the vitamin D specifically, essentially what my supervisor had found, she's an amazing woman who's a kind of leading scientist at the company. She essentially utilized and tested different wavelengths of UV. And if we just expose the mycelium to UV while it's still alive before we go to final processing, it can create vitamin D at an insane concentration. We got it to be like 2000% of the daily value accidentally. And then we said, okay, let's ratchet it back. We're actually making too much vitamin D, and we want one serving to have, say, 100% of your daily value. So it's something where mushrooms obviously have that capability, but the mycelium of this fungus that we grow has that same ability as well.
Yeah, you know, I grew up, you know, when eating vegetarian was fairly a young thing to do, and you literally had to combine foods, like you had to combine, you know, corn and beans in order to get a complete set of essential amino acids for, you know, completed protein that the human body needs to have. And it would seem to be a logical thing if you could possibly do that, you know, in one product, like the mycelium, to tweak it in such a way that it is a complete source that doesn't require, well, you got to put this in with it in order to make it work for you.
That, so, by the way, is always been about the other amino acids, and they found in body stores. That changed. Because the diet for the lonely planet, cookbook used to say that, and they really yanked on it later on.
Well, that's what I know. We are back in the day.
Right, back in the day.
Yeah, I mean, you know, the whole,
way back.
You should not, Rob.
So, well, actually, it goes back a lot. It goes back a lot longer than that, because the Native Americans were intentionally combining these different foods, the corn and the beans, and they even treated the corn with lye to convert it to the right formula so they could get a complete nutrition.
That's super interesting.
Yeah.
I definitely didn't know about that.
Yeah.
So I guess we will keep chugging along. I definitely want to try and get through the rest of my material that I prepared today. So I do want to talk about these fungal raw materials now explicitly. We're going to talk about some actual products that you can either find in stores or that are being made to be sold by these really amazing and cool companies. So I just wanted to put up this big report right here. It has all these different companies, and I'll just talk really briefly about all of them. So in the top left, we have Ecovator. They're really one of the most nimble and companies in the space right now. They're making a bunch of different products with mycelium. They're making a styrofoam replacement. They're making meat in the form of that mycelial bacon. They're also making things like acoustic paneling and building blocks too. So they're really doing a lot. I highly recommend checking out any or all of these companies that you feel interested in. You can Google them and things like that. They're in the news. MycoWorks, I used to work there. They're making the mushroom leather. They're based here in Emeryville, California, and they just opened a factory in the East Coast in South Carolina. We have Bolt Threads. They're also very much focused on the textile dimension of this, and I believe that they were originally doing some work with the spider silk and the goat microbiome and this crazy work with the spider silk, and they kind of have branched into the mycelium materials as well. Nature's Fine, they're making food with mycelium. At Last has actually been rebranded since I last made this. I didn't get to update it in time, but they're now My Forest Foods. They're a subsidiary of Ecovative, making the mycelium leather. Mycelium bacon. Mycorrhiza is a great company out of Sweden, and they're making mycelium food through liquid fermentation, similar to my company. We have Mogu, based out of Italy, making acoustic soundproof panels with mycelium. And then finally, Corn and Meaty, very much doing that similar liquid fermentation to make a food product. So all these companies, once again, corn is in stores, you can find it everywhere. Meaty, as I was mentioning, can be found in Sprouts grocery stores. So many of these things are getting out there. You can buy, I think, like hats and jackets from MycoWorks made out of mushroom leather. So we're finally getting over the cusp of conceptual ideas to products in the market that people who aren't absolute millionaires can buy. So that's a very cool accessibility thing. And so I'm going to do a little bit of a deep dive on a few of these. I want to talk about Ecovita, MycoWorks, Corn, and Meaty, because they're some of the companies that I know the most about, and then I think are just great examples to look at as far as what they're doing. And so I just want to show once again some visual examples. The left side two photos are going to be Meaty. We have a steak and a chicken product. We have some of those that you'll be able to try shortly. Maybe just as soon as we wrap up, we can kind of like have a tasting and just all check it out together. We have these really interesting sculptures that have some Ganoderma fruit bodies coming out of them. So it's like this is more of an art application than a practical, but still very cool and inspiring. We have those acoustic panels from Mogu. We have another art installation with these kind of almost hides that have been grown with these very kind of natural undulating shapes of mycelium. And then we have that kind of mycelium bacon that I was mentioning. So there's really so many things that are emerging that if I showed you these left side two photos, would you think that that's mycelium? I mean, that looks pretty convincing to me. So we're really able to produce a high level of reproducibility for a product that maybe was irreplaceable before. So looking at some of these practical mycelium materials, we can kind of talk about a little more in depth what are companies doing. So MycoWorks, as I mentioned, I worked there with Will, who's sitting here in the front row. We started on the same day and shared a journey there at that company. And it was a really incredible place to work. So they were making fine mycelium. At the time I worked there, they were utilizing a Ganoderma fungi, and they coined the term made with reishi because they're using that reishi mushroom. They're making handbags, they're making a headphone case. We have an example of a hat. And then once again, back to the kind of artistic and sculpting aspect, this is a bowl of fruit made out of mushroom leather. That's just kind of an interesting art piece. So they're really kind of trying to bridge the gap between fashion, art, sculpture, and that's all due to Phil Ross, who's the founder. So I went ahead and put up a picture of Phil Ross here. He's been working with mushrooms and mycelium for over a decade, and his conception for this company is, I can perturb this mushroom, I can treat it in a way that generates a unique material property, and that is what he turned into the mushroom leather product. So he did a lot of work with sculpting, he did a lot of work with these building blocks and things like that, but as you can see, they've kind of jumped into mushroom leather, targeting high fashion, and they've partnered with Hermes of Paris to make these really high quality handbags that they make the Birkin bags that sell for $50,000. And so they're just targeting the luxury industry to break in, and then they want to expand and grow outward from there and kind of top down mentality. We have Ecovita next up. Once again, I mentioned them earlier. They're just doing so much in so many different realms right now. So like I said, they have that mycelium foam on this left side with some perfume in there. We have mycelium leather, mycelium bacon, shoes and things like that. We can really leverage these products for really whatever your mind can conceive of. I mean, if you can dream it, we can make it a reality in some fashion, I'm sure, because there's so much innovation. And so this is some of their mycelium, I believe, and this is kind of pretty common knowledge based on their publications and the videos that they post. So that My Forest Foods product, you see these kind of mycelial kind of rods on the left side, or these kind of long, fluffy looking marshmallow looking things. That's the aerial hyphae of that organism. I don't know exactly what species, but they grow this fungus, they get the aerial hyphae, slice it at the bottom, they can now turn that into their baking product, and that's how they're getting this unique texture that maybe isn't 100% baking quality and with the crispy and the crunchy, but it's at least pretty darn good. Like, this is probably some of the best vegetarian bacon you're gonna find, I would wager, because mycelium is so unique and cool in that way. And then finally, more of the styrofoam product. So really, if we can eliminate styrofoam with mycelium, you use that product, you get your package in the mail, you can take that styrofoam mycelium block, throw it into the yard, that's gonna compost, and you don't have to worry about it. So could be really impactful. So that's some of the materials. I'll go on to talk about some of these foods as well. So, you know, here's my company, Meaty. We're making whole cut alternative meat using fungal mycelium. We're different from someone like Impossible Foods and Beyond Meat. They're making a lot of these ground meat products. They're not super substantial. Sausage, meatballs, that's great. Where's my steak patty? Where's my chicken cutlet? Where's my actual substantive meat product? And that's what media is trying to do. So once again, we do liquid fermentation, we do minimal processing, add some ingredients in there, and we get these really awesome, tasty stuff at the other side. Not exactly. We do target the meat and umami flavors, but we're not using heme, for example, like they use. We're kind of just going for plant-derived spices that we can just kind of add in there to give a very rich and umami flavor. And so kind of as I've been mentioning, liquid fermentation does have unique challenges. Those are the kinds of things that we try and workshop and deal with every single day, putting out a lot of fires, starting up a factory. But our factory is up and running in Thornton, Colorado. It's 80,000 square feet. And the meaty product is nationwide. We're in every single Sprouts in the United States. So if there's a Sprouts near you, I know there's a lot in California. I don't know if there's any in Santa Cruz specifically, but there are some in San Jose, up on the peninsula and stuff like that. So you can find us in stores, and it's really great to see a startup going from conception to reality, going from an idea to in the actual market. And we're sponsored by celebrity chef David Chang, if you've heard of him. He has the Momofuku restaurant and a few other business ventures. Really cool chef who has featured our stuff and has special recipes using the meaty that you could make at home. So it is a really cool thing. And then corn, I cannot not mention corn because they've been doing this exact same thing for decades. And it's actually a super interesting history. I highly recommend, if you're interested or haven't heard of them before, to look into the history. They were started in the 1970s to develop a fermentation process to address protein shortages. People thought that protein in people's diets wasn't going to be sufficient, coming off the back of several kind of world conflicts and world wars and things. They were just so worried about the food stability and food security. And this is kind of at the same time as chemical fertilizers were coming online. We're kind of getting agriculture, more dialed in at a global scale. And so those protein shortages they anticipated never really hit, but this company was still able to grow and develop a unique process where they take a Ascomycete fungus, which is the Fusarium venenatum, they do the liquid fermentation and they make their product. So they're in grocery stores as well. They have chicken nuggets, they have this cottage pie, a lot of they do mincemeat, right? It's very British types of food, but it's great and highly recommend to try it as well. So this is kind of just another workflow diagram. They have a lot of processing they need to do to take their food to market. They found their organism, they're able to grow it. It has these super interesting little croissant-shaped cells that are going to be grown, and that's how they make their food. And I kind of wanted to zoom in on the bioreactor. So a little bit more on the technical side, but this bioreactor is so unique because they're able to do an airlift style reactor, meaning they're kind of bubbling air in from the bottom. The media is fully turning over and going over the soil to circulate, and then they're continuously harvesting from it. So every 6 hours, 12 hours, whatever that interval is, they take mycelium out, they put some media in, what's still in the reactor remains in there, and they're consistently cycling to harvest this material. So it's something that kind of just runs 24/7, and they're able to get this huge efficiency. So there's kind of a lot to dive into if folks are interested, but it's just a very unique design. It's a cool product, and they're just absolutely going for it in the UK. And then this is a more kind of traditional style bioreactor. So not to belabor everyone with the jargon, it's kind of a very detailed diagram, but you essentially need to have airflow coming in, airflow coming out, maintain sterility, mix the liquid, dose base in, because as the organism grows, it becomes more acidic. So you want to dose base to balance that pH, keep it at a good pH for growth. Don't want it to kill itself by growing too much. And so there's all these things to consider in fermentation in these processes. So that's a lot of my content. I can pause here and I just have one small section to the end. So maybe we should just hold questions and do a full Q&A and open up this discussion. But I want to talk about the future of biomaterials and sustainability, because this is an exciting time. Companies are doing so many different things, and we're discovering so much about mycelium and fungi and ecology that we didn't know before. But at the same time, the future can be kind of scary. I think it's hard to think about things like global state change, climate change, loss of biodiversity. All these things can be really harrowing to think about and can be almost paralyzing. But I hold a lot of hope because of the innovative work that people do. And I see these solutions on the horizon that biotechnology is going to enable for humans and for society. So just to kind of go through my spiel really quick, this is kind of my concluding notes. Naturally derived biomaterials are showing us a near infinite source of utility, innovation, and inspiration. They can disrupt industries that are mired in old ways of conducting business and profiting off of the exploitation of the environment and of the people that inhabit it. We're really only at the start of this whole process of understanding ways to utilize nature to sculpt and mold these natural processes to give us a desirable result. As we learn more, material properties and strengths will be improved, and all these different biomaterials will really be at par, if not better than their existing counterparts. I really want to see alternative meat be more desirable than meat. I really want to see alternative leather be stronger than a cowhide, because you're not going to convince people to say, hey, you could try this different thing and it's as good, but it's different. But if you instead say, hey, this thing is way better than what you're used to, give it a try, this is how we're actually going to drive societal change and drive people to change their buying habits in the marketplace. So I'm very excited about that. And I really do envision a future where living materials are the norm and that sustainability is inherent in the workplace and in industry, because we cannot continue to exploit the environment and it will not support us if we do. So we really need to move towards sustainability using these new ideas, using industry, and using a kind of harmonious way of interacting with nature. So that is my spiel for today. Thank you so much for listening. Yeah, thank you so much. And I'm happy to take any questions that you all have about really anything that we talked about, kind of a lot, but happy to go in depth anywhere. Yeah.
Does your company do anything in the way of seafood products, fish substitutes or shellfish substitutes?
My company doesn't currently. We have thought about it. We've definitely experimented and tested a few kind of fish formulas that we've tried. There are a lot of companies who do focus on that. Yeah. There's one in the Bay Area called Finless Foods. And I think the difference there, or the kind of different strategies are, do we want to make a plant product and make it taste like a fish? Or do we want to take a fish cell and grow it similar to like lab grown meat, cultivate those fish cells and kind of grow a fish fillet? So those are the two things I see out there right now. And there are a lot of companies, but mine currently not focusing on it. Yeah.
What's the impact on climate?
Yeah, that's a super good question. And I would say it's just really, I can repeat the question. So the question was, what's the impact on the environment to produce all the chemicals and the ingredients that are in the liquid media for these fermentation processes? I would say the answer depends on the media and it depends on the scale as well. And so it depends on, once again, are you using something that might be, for example, an agricultural waste product? So corn steep liquor, for example, is a byproduct of corn, and they kind of take all these corn hulls and things like that and ferment them and get this kind of product out the other side. So that's an example where your entire media could just be corn steep liquor and you're kind of taking this agricultural byproduct. So I'd say something like that has a minimal impact. But if you're going to the process of saying, oh, I'm going to have my defined media with all these discrete chemicals that are made, you kind of have to go and track, well, how is the citric acid made and where is that from? And where is the copper taken from, and how is that, gotten to me, and who's generating that product? So it's definitely very nuanced, but I think as companies grow, and even a company like mine, we are trying to double down and look at our carbon footprint to ensure that every part of our supply chain is sustainable, or at least as sustainable as we can get it to be. And so I think it's on the burden of the company, and as unfortunate as it sounds, to be tracking that for their own supply chain, because you want to get your product from a reputable source and from a source that isn't just mucking things up, essentially. Yeah, and then, yeah.
And other things that might be harder to
Yeah, that's a super good question. What I can say to that is all the things that we're putting into our media for something like liquid fermentation are things that you can find in nature in some form or another, and they're all in a bioavailable format. So everything we're putting in can be metabolized and digested by the organism. And so it's almost going through a filter before it's even getting to you, right? The mycelium itself has to grow. We're not necessarily keeping that broth into our product as well. So when we harvest the mycelium, the broth is left behind, it's drained out, it's rinsed and things like that. So nothing that's residual in the media is coming into the product. And what you're getting is just the mycelium itself. So you can think about harvesting a mushroom from the forest. It's obviously gone through its own ecology. It's had to digest nutrients in the soil and things like that for it to grow. It's kind of just an analogous process, but in a different system and in a different place. But I think maybe the most important point that I can say is like, everything is in a natural state, but it maybe is just distilled into a crystalline form, for example, then put into the liquid to feed to the organism. So everything is going through the organism, and there's probably something to be said for doing long-term studies on the impacts on health and on the digestibility. But we've gone through FDA approval, we have grass approval, so generally recognized as safe. And corn has been in the marketplace for over 30 years, and people eat that mycelium every single day. Some people do, and there's been no kind of detriment that I've seen from that. So I think that that's maybe our best example of like a long-term product using liquid firm. It's a ascomycete that we can just eat it and be fine. Yeah. Okay. Yeah. Right. So are you saying that it's desirable to remove it from there? I'm curious about exactly what you're asking. So are you saying, is that something where we want to take that out for a reason? Or are you curious about just learning more about it? Or I'm not exactly sure what you're asking. Right. Okay. I see now. I think that the mycelium in compost is definitely desirable. I think it's going to be a very complex kind of biological system where you have many bacteria, many fungi. Oh, sweet. Many fungi all together. And so there's probably a lot of diversity there. But I also think that diversity is a good thing. And we could stand to say, take some samples from there, culture some of those fungi and bacteria, and they could probably be used for a purpose. And they probably have a unique function that we might not even know about. So I think that diversity is good. Having a healthy compost is definitely desirable. And some people do study those microbes that are all in there working together and how do they relate. So it is just a system that we could study. And those are organisms that you could grow for a purpose and maybe make a biomaterial out of any one of them probably. Yeah.
You have a chat question.
Oh, yes.
Adrian Pollock.
Oh, Adrian. Are there okay, I'll read the question out loud. So are there companies who are doing genetic engineering of fungi for biomaterials and foods? That is an awesome question from my friend Adrian on the chat. So the answer to that is yes. There are companies doing genetic modification of fungi. It's not particularly easy to do. It's something that is still being very much developed. My company does not. We have a food product that we do not ever want, or at least right now, to ever have labeled as genetically modified. The public doesn't love that. We as the scientists aren't opposed to it, but we don't want to upset the public and have a product that everyone's afraid of. So for the time being, we don't need to GM. It would definitely make our lives easier, but we just can't practically do that. Companies that make non-food products are probably much more likely to use GM. If you're using mycelium for leather and you have a GM fungi in the leather, I think some people still might be scared of that, but people will be less scared of that than a GM food that you eat. And so the answer is for sure yes. I wouldn't know exactly which and exactly how, but definitely GM is a solid method for getting the material properties better or getting it to grow faster or do a different function for you. And so I think maybe we can take one more question and then we have our meaty that is cooked and ready to go. So we can maybe transition to trying some meaty, having a more walk around kind of time and close out the lecture session. But what's the question? Nice.
So my question is real basic. We grew up foraging mushrooms, and we know that mushrooms have a chitin on the outside, and we tell people you have to cook the mushroom. Otherwise, as far as nutritional value, you're not going to get the nutrition out of the mushroom unless you cook it. With the mycelium grown in either a solid or a liquid state, is that cooking still managed? Is there still chitin there?
Definitely, yeah. So chitin is going to be found in most every true fungi, to my knowledge. Someone, if they know better, can definitely jump in. But I think you're going to find chitin in a lot of these, or if not all of these fungi. A lot of the companies that are making the food are going to be selling it in an already cooked state. So, for example, BP sells an already cooked product that you essentially just have to reheat, or you can obviously cook it again as well and add your spices and flavors and what have you. But we do a cooking process to ensure that, for one, it's dead. We actually don't have a kill step that's not cooking it. So that's our current way of just arresting the biology. But then additionally, we then just go to freeze it, lock in the flavor, make sure it's not degrading or anything like that. And so in a lot of cases, it's either going to be sold cooked or you will be advised to cook it. I don't know if there's a product out there that someone's selling, for example, raw mycelium. And so I think for a lot of these, corn as well, it's already cooked. They do that at the factory and then sell the product. So for a two-part answer, yeah, there should be chitin there still, but it's already cooked, and you can also cook it yourself to kind of ensure that it's been fully prepared. Yeah.
There's a drink that's like mycelium. I don't think they're doing that great.
Oh, interesting.
I've seen private report. Beer's not allowed. But
it's like that for health kind of thing.
Yeah.
Okay. Gotcha. Super interesting. Yes. Thanks, everybody. Let's check it out.